Can you see the okay, recording progress? Great. So, good morning, good afternoon to everyone, and a very warm welcome to all the participants joining today. On behalf of Paleo ECR India and the event coordination team of the Paleo ECR India community, I'm very much delighted to welcome you all to another session of the Paleo ECR India Saturday Talk series. Actually, this actually this initiative was started with the goal of building an open and interactive platform where early career researchers, the senior scientists, and like other students can come together to exchange the ideas, discuss the new scientific, findings, and promote the collaboration across the diverse field of value science and earth system sciences. So today we are pleased to host Dr. Shubham Tripathi, who is currently serving as a principal project associate and NABL technical manager at the Sophisticated Analytical and Technical Help Institute, SATHI, BHU, India. He completed his PhD in marine science from National Center for Polar and Oceanic Research, Goa, in 2021, and previously worked as a SERB National Postdoctoral Fellow at BHU and as a project scientist B at NCPOR, Goa. Research focuses on paleomonsoon variability, climate change, denitrification. Mostly, he has published a very good paper in the Arabian Sea denitrification work. Stable isotopes, carbon, nitrogen, oxygen, magnesium and calcium measurement in the forams for estimating the temperature. He has a good expertise in IRMS, ICP-OES, and now currently he is handling the HR-ICP-MS in SATHI BHU facility. The important thing is he has also participated in IODP Iceberg Alley Expedition 382, and it's one of the rarest opportunity people get. So, and also contributing to the studies of past ocean climate interaction. So today, the title of his talk is Measuring Past Ocean Temperature Variability, Techniques and Uncertainties. So before we begin, I would like to just share a few announcements. First is its request to all the please keep your microphone muted throughout the talk. Please feel free to post your questions in the chat box or use the raise hand feature. And the presentation will be approximately 40 to 45 minutes. And after that, we will have a separate 15 to 20 minutes Q&A session. I request all the participants, all the ECRs to participate in this Q&A session and discuss as much as you can. Your suggestions are very much valuable for the speaker as well as the value in the community. So with that, it's my pleasure to invite Dr. Shubham Tripathi to deliver today's talk. Thanks, everyone.
Thank you, Arvind, for inviting me to present my work here on this forum.
You please share this.
Okay.
Perfect.
So I think my slides are visible to everyone.
Can you just make it like a PowerPoint mode here, presentation mode?
Yeah, this is in PowerPoint mode.
Okay. No, it's clicked on somewhere else, like it's not on the.
Okay. I think now it is in PowerPoint mode. So
Yeah.
Thank you everyone for joining this talk. So I'm very excited for presenting the measurement of past ocean temperature variability And I have made the talk in such a way that I will talk about why measuring past ocean temperature is important and what are the techniques through which we can measure the past ocean temperatures. And I will focus more on the uncertainties related to those measurements, because we do a lot of interpretation based on the measured temperature. So we should know how much uncertainty is associated with those measurements. So right now I am in SATHI. It is a NABL certified lab with certificate number 12810. So NABL means National Accreditation Board for Certificate and Certification and testing laboratories. So it provides certificates to the lab which do different kind of testings and calibrations. So SATHI is one of the NABL certified lab. Yeah. So when we say that we want to measure something, some climatic parameter, and we are able to assign numbers to that, it is known as quantification. So by measuring different parameters of climate change, we can do the quantification of climate change. So measurement, analysis, and numerical expressions of the variation in the Earth's climate system over time is quantification. And different factors are there, different indicators are there which can be measured, like thermal indicator, atmospheric indicators, relative and energetic parameters we can measure. We can measure the extreme event statistics, like how many cyclones or how many low pressure systems has taken place in some kind of basin like Arabian Sea or Bay of Bengal. So we can measure different kind of indicators and we do the quantification. My focus will be on the thermal indicators, which is sea surface temperature. And when we say that sea surface temperature, is it the surface temperature, or we measure the water column temperature at different depths? So this will be the focus of my talk. So moving further, what does ocean temperature? Why does ocean temperature matter? Why do we want to measure the ocean temperature? What is its climatological importance and atmospheric importance? So let us say what IPCC has to say for this. So IPCC fourth assessment report shows that the observed land air temperature and sea surface temperature trends are consistent globally, with land warming more rapidly than oceans. And this underscores the physical link between ocean surface and air temperatures. And then the sixth report also discusses how ocean warming drives changes in the atmospheric circulation and contributes to regional patterns of land surface temperature change. The sixth report emphasizes that changes in the ocean heat content and SST have far-reaching effect on the climate over climate extremes and atmospheric temperatures. So these are the changes in which we are more interested, climatologists and atmospheric scientists. But are there any other effects which are more realistic, which we can see in the present day and which we can realize more than the climatology and atmospheric science? So I have gone through some papers for preparing this talk. So this is one of it, and it is about to understand the role of sea surface temperature forcing for variability in global temperature and precipitation extreme. This is by Dittus, and it is one of the latest outcomes for sea surface temperature measurement. Then another paper which talks about global and regional evolution of sea surface temperature under the climate change, how sea surface temperature is evolving in the present day climate change. Then increasing rate of global mean sea level rise during 1993 to 2014. So this paper talks about the recent happenings in the global mean sea level rise due to the sea surface temperature change, or we can say ocean water column temperature change. This is very interesting. This talks about global patterns of sea surface climate connectivity for marine species, how marine species are affected, and it shows very realistic effect of sea surface temperature and climate change, global warming. Another paper which talks about key indicators of Arctic climate change from 1971 to 2017. So all the changes which take place in Arctic and its indicator are discussed in very much detail in this paper. So you can go through all these papers if you are interested. and another one which talks about the global vulnerability of marine mammals to global warming. So we can see that marine fauna is very much affected by the global warming, and a lot of papers I have gone through for preparing this sea surface temperature talk, because it's always cliche that when we say that sea surface temperature causes this, like increases the intensity of cyclones and all those things. So let us see some realistic things. Look at this map. This map shows the pixel classification based on the thermal analog condition. So we have read about the thermal analogs, climatological thermal analogs. They're like present day thermal analog. This mid-Pleistocene warming period is the present day analog in the past of the climate. But this shows the surface analog on the ocean surface. So you can see different colors: pink, orange color pixels in the Arctic, in the Pacific Ocean, blue pixels over the Indian Ocean and Southern Ocean. So, this talks about the surface climate connectivity. So, when temperature changes on the surface in one region, the organisms, the fauna living there, they try to move to some other place where they find their uh, uh, the temperature which is in which they were previously living. So that particular region become thermal analog for those animals. So I came across very interesting term. It is known as climate velocity. So the direction and rate at which organisms must move to maintain a given climate is known as climate velocity, and it is a vector term. Means it has both velocity, speed, and direction. So in this map, if we see, pink will not have a thermal analog and will not host past thermal conditions in future. So all the area where you can see pink pixels, they don't have thermal analog. So if the temperature rises in these regions, they don't have any thermal analog. So species living there, they are endangered and they might get extinct in future. And all the blue pixels will have thermal analog but will not host past thermal conditions in future. And orange will not have a thermal analog but will host past thermal conditions in future. And whites will have thermal analog and will host past thermal conditions in future. So this paper is from Petsas 2022, and he has discussed in very much detail about this thermal analog thing and how it affects the marine organisms. Um, so they have discussed that a longer trajectory may be required by species living in the northern hemisphere as compared to in the southern hemisphere. And in the northern hemisphere, the species which are living in the northern hemisphere, they are more prone to the exposure to the climate change and extreme conditions. And more than 27% of surface ocean have no thermal analog, indicating the unique climatic conditions would be lost by the end of this century. So we can, this paper help us to realize that how it is important to measure the sea surface temperature and know it accurately. And if we can go in the past, then we can try to reconstruct these kind of thermal analog in for the past time also, that whether some species has migrated in the past or they have changed their thermal regions, how the climatic velocity has changed. So this is all about the introduction that why thermal measuring the sea surface temperature or water column temperature is important. Now we have to know that how we can measure, what are the techniques through which we can measure the sea surface temperature or water column temperature. So let's move further. So what does measuring temperature in the past really mean? And what does SST proxy really tell us? When we say that we are measuring SST, very often we say SST. But let us say if if I choose G seculifer, it resides in mixed layer region. So it gives you mixed layer temperature, not the SST proxy. So it is very important to know that what particular proxy we are choosing, what species we are selecting, and what kind of temperature, what kind of climatology we are able to reconstruct using that particular proxy. So all those things we discuss in the later slides. So this is very good book by William Rudiman, and I have kept this here so that we can read about the textbook things like what is climatic sensitivity, what is proxy, what are climate archives. All those things are given in very interesting way in this book. So I recommend this book to everyone who is seeing this talk. Moving further, different paleoceanographic proxies capture different aspects of temperature variability and in general different aspects of the climatology. So species living at different depths, they will give temperature for different depths of the water column. Different seasons can also be measured by selecting particular species or particular proxy which are sensitive to a particular season or, at particular depth. Different ecological environments can be captured by using selected proxies. So reconstructing past ocean temperature requires understanding of what component of the temperature we want to measure, and we have to use our wisdom which proxy we have to use and which species we have to select. And also very important is identification of the species. Let us say if you are if you want to measure saccalifer, if you want to use saccalifer, then we must have full knowledge how to identify saccalifer, bulidus, or ruber, and then the data generated from that particular species is reliable, and we can talk about different kind of climatologies related to SST or mixed layer temperature or bottom water temperature, etc. So different kind of proxies are there. So planktic foraminifera is one of them. Then text 86 is there. Different kind of organic proxies are there. Diatom is there, and we can use assemblage studies. We can use artificial neural network to reconstruct this temperature. So planktic foraminifera oxygen isotope was also used for reconstructing the temperature. But we know that oxygen isotope ratio in the cell, it depends upon both temperature, isotopic composition of seawater, which is known as ice volume effect. So the oxygen isotope of 4 amps, it is not only affected by the temperature. There are many other non-thermal effects which affects it, and the data which we get from by using the temperature which we get by using oxygen isotope, it is not as reliable as many new proxies which we have already generated. So moving further, this work was first done by Emiliani 1955 and Shackleton 1967. So they have used oxygen isotopes to, generate sea surface temperature. So we can use mg by Ca ratios. That is, these are the trace metals, and we can use the mg by Ca ratio for reconstructing the sea surface temperature. And as the temperature rises, the magnesium ions, they replaces calcium in the carbonaceous cells. And by many workers like Leah et al. And some recent papers also say that per degree Celsius, 10% increase in magnesium takes place in the cells of the foraminifera. So all the proxies have their advantages and their disadvantages. Some of the proxies, they are very reliable in polar regions. However, some are very workable in tropics. So sometimes this becomes their, advantage, and sometimes it becomes their limitations. So mg by Ca ratio, which is measured in the 4 amps, it is very useful in tropics and subtropical regions. And we can measure temperature for the whole water column, maybe surface, upper mixed layer, lower thermocline, upper thermocline, mid water depths, and even by using benthic forums, we can reconstruct the temperature of the bottom water. They are highly sensitive to the warm temperatures. So the range of mg by Ca in many texts, it is from 14 to 30 degrees Celsius. So this is the range. So below 14 degrees Celsius, the data by mg by Ca ratio of 4 amps, they are not reliable, and we can shift to some other proxies. So for knowing geochemistry of forams, you can refer to this talk by Dr. Sambudh Misra. It is on the platform of Kusman Foundation. Very detailed talk on the different kind of geochemistry of forams like boron isotope measuring boron and MgYC, and everything is given there. So these are some of the important papers which I have referred, and they have done different kind of analysis for measuring MgYCA and how MgYCA is transformed into temperature. So limitations of MgYCA is dissolution and salinity effects, and its performance is not gets unreliable below temperature 15 degrees Celsius. So how this mg by Ca is converted into temperature, you can refer all these papers to know that how the empirical relations equations have been generated. So sometime the culture studies and species specific equations are there. Mixed species equations are also there. So in different scenario, we need different kind of forams, different kind of species. Let us say you want to measure sea surface temperature or mixed layer temperature, and you don't get any uh, single species in a quantity so that you can measure mg by Ca. So in that case, you don't have left anything, so you have to use mixed layer equation. So every equation have its importance. And let us say in my study, I have used G cyclifer. So G cyclifer was the only abundant planktiforum. So I have used that, and I have reconstructed the mixed layer temperature of Arabian Sea. So some of the latest paper by Dissard et al. 2021 and Nairn et al. 2021. So Dissard has included strontium by calcium also in the same equation of magnesium by calcium. So I will attach the link for this paper, and you can read in more detail that how they have modified to get more reliable temperature, sea surface temperature, mixed layer temperature by using planktiforams. And in general, the error associated with mg by Ca paleothermometry is around 1 degree Celsius. So the next proxy, these are some of the useful papers. You can go through this user guide for choosing planktiforams, mg by Ca temperature calibrations, then constraining multiple controls on planktiformifera mg by Ca, whether it is affected only by temperature or some other effects are there which affects mg by Ca incorporation in the calcium carbonate of planktiforams. Mg by Ca and SR by Ca ratios of planktiforamifera, proxies for upper water column temperature reconstruction by Clearux et al. 2008. So yeah, now we move to the another proxy, alkenones. So these are the long chain unsaturated ketones produced by haptophytes, alga. The degree of unsaturation ratio of di to tri is highly temperature dependent, and this is being utilized to reconstruct the water column temperature. So they are suitable for tropical to subtropical SST reconstruction, tropical Indian Ocean studies. So UK means alkenones SST estimates may underestimate SST where the temperature exceeds 28 degrees Celsius. So this is the limitation of this proxy. If you want to measure the temperature where sea surface temperature or water column temperature exceeds 28 degrees Celsius, so the data from this proxy is not reliable. These are some of the papers in the bottom. You can refer to get the more detailed studies and the equations to how to get temperature from alkenones. The next one is diatom. Diatom biomarker proxy. These are the highly branched isoperinoids, also known as EZ25. This is one of the latest proxy developed by Simon T. Belt group. And this is one of the latest paper. It came in 2025, and this group has published a lot of paper based on diatom lipids and diatom biomarker based proxies to reconstruct the water column temperature in the ocean. And it is calibrated over -1 degree Celsius to 18 degrees Celsius. So mostly this is useful in Arctic and Antarctic region where the temperature is very low in this range. So similarly, lot of proxies are there, like clumped isotopes, ANN techniques you can use. Uh, you can use TEX86. You can use diatom biomarkers. So these are the tabulated. This slide show the tabulated form. Which proxy is useful in which particular range and in which particular region. So you can refer this slide to get the idea that text 86 can be useful for -2 to 30 degrees Celsius. It works globally and often useful in giving the subsurface signals. Mg by Ca is not very reliable proxy in polar regions because a lot of dissolution takes place in polar regions and CO2 is much dissolved in polar region water. So it corrodes and you know, it corrodes the surface of the forems and that effect will definitely reflect in the Mg by Ca data in those regions. So better to use some other proxy which is more reliable for the polar regions. So this is all about the proxies. And now I have made a conceptual model where we can see a parameter to a proxy mapping. So let us say you have species 1, which is living at the depth 1. Let us say it is surface. And there are 4 species and all living in the different depths. And you want to measure temperature at all these depths. And let us say you want to measure at the surface and the species 1 is not there. So can we use the species 3, which is living at the depth 3 and species living at the depth 4? Can it be utilized for measuring the surface temperature? Or can species 1 or species 2 or species 3 be utilized to measure the temperature at depth 4? And they are thriving at the different depths. So yes, Mg by Ca versus temperature is kind of mapping of these proxies. So surface species can be utilized to measure the temperature at D3 or D4 at the deeper levels. But in that case, error will increase. So you have to generate the time series for, let us say, species 4, Mg by Ca time series, and the temperature of the surface. And you can make an empirical equation for species 4 to the surface temperature. Similarly, you can make an empirical equation for species 1 to the depth 4. But the error will substantially increase. So this is one way to look at these proxies. So in case when we have species 4 at depth 4, this will give you the most reliable data. So this is one way to look at the proxies versus parameter construction. So how do foraminifera record temperature? So I will discuss a little about my PhD work, and then I will move towards the main part where I will discuss about the uncertainties related to these measurements. So the composition of their calcite cells depend on environmental conditions, pH and all those things, temperature when they grow. Temperature is recorded in their cells by the amount of magnesium present. So Leah et al. And all those paper have given the quantification that how that every 10 degrees Celsius there is every 1 degree Celsius there is 10% change in magnesium. I have used G Saglifer for measuring mixed layer temperature in the Arabian Sea. These are the same images which I took during my PhD from NCPOR scanning electron microscope. So why I have used G Sacrifer? So one thing I have already discussed that it was the most abundant in most of the samples I have used. In general, it has high relative abundance in the northern part of the Arabian Sea, mostly greater than 30%. These are some of the references like Bay 1977 and Naidu et al. 1993, which talks about their abundance in the ocean. G sacrifices spinos mixed layer dwelling planktiforms. So if I want to reconstruct the mixed layer temperature, I have to choose G sacrifier. There are many other species also, but it was the only abundant species I found. And it goes from 25 to 40 meter. It lives. This is the range of thriving of G sacrifier. And G sacrifier is also resistant to dissolution compared to other planktic foraminifera species. So this is a relative sentence statement that it is resistant compared to other some other species living in the mixed layer. So this is another picture which talks about the range in which G. cyclifer thrives. It is from 17 degrees Celsius to 30 degrees Celsius. So it is very, appropriate species to work on in the Arabian Sea, because the Arabian Sea temperature vary in this region, in this range. This is another probability density function for the calcification depth of different species. And this is from Farmer et al. 2007 paper. So this shows that Cyclifer is lying here. So you can see that this water depth is from 20 to around 45 meters. And this is the probability density function. Yeah, so I have used Dickens et al. Equation for getting the paleo temperature in the Arabian Sea. So this is very important slide for MG/CA measurement. So as of now, we use Barker et al. Modified protocols to clean and remove the impurities from the foraminifera. So around 15 to 20 tests I took for measuring MG/CA from one sample. So we have to remove the impurities, organic impurities, and let us say secondary carbonates from the plankton foraminifera, because we want to measure Mg/Ca from the carbonates of foraminifera only. So we have to remove the other impurities. So how do we know that we have removed clay and all those things? So for that, we measure iron by Mg ratio, Fe by Mg ratio, and it should not exceed 1 mole by mole. And if it is more than that, those samples will be rejected, and it is considered that they have considerable or substantial amount of impurities, and we are not getting the signal from carbonates of the plankton foraminifera. So another important aspect of this slide is we have to have the regional empirical equations. We don't have, as of now, we don't have Mg/Ca based paleothermometry equation for Arabian Sea. So I have used the closest. So this is from Dickens et al. 2002. This is from Eastern Pacific, and the climatology and ocean atmospheric relation is very similar to the Arabian Sea. So I have used this particular equation, which is from Dickens. But if we have empirical equation from the Arabian Sea, it will give you more realistic and reliable data. So this is what we need to do for Mg/Ca measurement, and for many other proxies also. Regional measurement is important. So this has shown the repeatability of 0.01 micromole per mole, and standard solution was having Mg/Ca of 2.18 millimole per mole. So the error associated with this particular equation is around 1 degree Celsius. So we have to minimize the error. For that, we have to create our own equations for different oceans, and then we can compare that with the global equations, and then we can talk about the global climatology. So this is again another picture graph which shows the seasonal variability of SST and sea surface salinity near the site U1457. This is IODP 355 expedition site, and I did my PhD from this site. So you can clearly see that near the surface temperature is varying from around 27 to 28 degrees Celsius, 26 to 28 degrees Celsius, and this variability slowly decreases. So there is no variability in the temperature after 600 meter depth, and this is valid for sea surface salinity also. So let us say if you are having species which are thriving on the surface in the upper 100 meter or in the upper let's say 150 meter, you will be able to get the seasonal signals. Let us say some particular foraminifera species, it thrives more in summer, so you can get the summer temperatures and you can get the winter temperatures for these particular depths. But as we go down, we will get only water column temperature. we cannot demarcate or we cannot separate the seasonal temperature after that particular depth using planktiformifera. So there are some proxies like alkenones, haptophyte alkenones, and it is said that the SSTs based on alkenones UK37, they mostly reflects the winter temperatures. So many text, many references talks about that. And text 86, it is base temperature reflect the SSTs in the oligotropic summer environments. So there are proxies which will give you seasonal temperatures, and there are proxies which will give you the water column temperature, and it averages out the seasons. You cannot demarcate that. So I have used this particular graphs to do the interpretation of my data. So let's move further to find out. So this is unpublished data from my PhD. I have generated MgCA based paleotemperature of G. saccharifer, and it ranges in the mid-Pleistocene transition period. It is varying from 0.7 million years to around 1 million years. And these are the interpretations. These are raw interpretations. I have not yet published it, so I will skip it fast. I will read it for you all. So we observed that precision and insolation 15 degrees insolation at 15 degrees south matches well with the oxygen 18 of saccharifer and co-varied in some of the marine isotope stages during the MPT. During MIS 28 to MIS 32, the insolation 15 degrees south and precision both seems to vary with oxygen 18 data. MIS 21 to MIS 18, the amplitude of the precession and insolation 15 degrees south become low. However, during MIS 20, there is an increase in the insolation 15 degrees south precession as well as in the oxygen data of the saccharifer. We observed that during MIS 19, this particular reason for which I did not found enough G saccharifer also to reconstruct the MG, but we have generated oxygen data. The amplitude of insolation 15 degrees south and precision is very low, but we noticed that oxygen of the saccharifer follow the same trend in the early to mid phase of MIS 19. This record reveals and provide evidence that South Asian monsoon variability is driven by precision, and this data corroborates the finding of Bolton et al. 2013 and Jibri Giouris et al. 2018 paper, which is from the south of the equator in the Bay of Bengal. And the South Asian monsoon is sensitive to the summer hemisphere forcing. So summer hemisphere, southern hemisphere has a lot of effect on the South Asian monsoon, and some of the recent paper even by Harshit et al., Singh et al. 2025, 26. They have shown that very beautifully that how southern hemisphere has huge impact on the South Asian monsoon. So this is all about the measurement and how MgCA is used in reconstructing the past water column temperature of the ocean. Now I will move to the main part of this talk, the uncertainties, how the uncertainties will be reduced and how it should be measured. So as the requirement of NABL, that is a National Accreditation Board for Testing and Calibration of Laboratories, we have to have different kind of repetitive and repetitive analysis of the standards and samples, and we have to generate a validation method for our measurement. So this is the first part. This I have taken from the validation sheet which we have developed for our IRMS data. So the first step is the suitability for the measurement, that whether our instrument is able to measure particular proxy or not. So we need to have more than 2 analysts to measure the same standards many times. Repetitive analysis takes place, and then we have to calculate the standard deviation. So analyst 1 has performed like 6-7 times the analysis of standard ammonium, IAEA N1, ammonium sulfate. and analyst 2 has performed same repetitive analysis, and analyst 3. So 3 has performed, and we have got the standard deviation and mean value of the that particular standard. So the next step is we have to find out the carryover effect, whether some carryover effect is taking place or not. So for IRMS analysis, we utilize tin capsules. So we have to measure blank tin capsules for IRMS. So this kind of work or this kind of practice can be performed for ICP-OES or HR-ICP-MS or whatever instrument you are using. You have to carry out these kind of practice to get NABL certificate, or to get a very reliable data. Then the third step is accuracy. Again, we have to measure the same standard or same samples at different days. So analyst 1, analyst 2, analyst 3 performs, and then we have to measure the recovery percent, how much recovery is taking place. So for the standard provided by the international agencies, they provide you the standard deviation of their own measurements, and they provide you the mean value of that particular isotope or trace metal. So we have to measure, and then we have to measure the recovery, how much we are able to recover from that particular standard. So this is step 3. Then step 4 is repeatability. Again, at the gap of some months, we have to again perform the same steps. So this is very repetitive and repeatable. But we have to do that to check whether our instrument is working well or not. This sounds very boring, but until and unless you do this, you cannot rely on your data for long term. Reproducibility. So repetitivity and reproducibility are two different things. Repetitivity means you have to run the samples or standard on the same day. And reproducibility, you have to run the standards at the gap of 1 or 2 months, whichever you decide. You have to make your own protocols. So you can see all the standard deviation, and they are in the reporting limit. Then ruggedness. So ruggedness is very important for the measurement. So in this particular case, let us say you are measuring nitrogen or CO2. If you are measuring the isotope of carbon or isotope of nitrogen, so you can vary the flow rate of these gases, CO2 or nitrogen. So for first measurement, you can take it at 65 ml per minute, 70 ml per minute, and 75 ml per minute, and you keep your carrier gas constant, and then measure the standards, and then find out how it is changing. Similar kind of practice can be performed for ICP-OES and HR-ICP-MS or other instrument. You can vary a particular parameter and then look at how your standards are behaving in those chains, whether your data is rugged or not. So there is no variation or effect on results by changing sample weight or the flow of the gases. So these are the way in which you can have a validation method, and you can check the uncertainty of your data. So this is the measurement of uncertainty, and this is by the guidelines of expression of uncertainty of Joint Commission for Guides in Metrology, and it is also based on International Organization of Standardization and International Electrochemical Committee. So this is based on the protocol number 170/25 2017 testing laboratory requirements. So for that, whatever practice you have performed till now, like you have done the ruggedness check, you have done the reproducibility check, you have done the carryover effect precision, all source of errors you have to measure. You have to make a checklist of all sources of error, whether it is coming from the your measurement from your pipettes or microbalance, all sources, their least count, all sources. You have to make a list of that, and then you have to measure the uncertainty. So uncertainty measurement is done by standard deviation divided by the under root of the number of replicates. So you will get the relative or individual uncertainty of one particular parameter. So you have, let us say you have 10 parameters. So you can measure the uncertainty for 10 parameters, and then you have to calculate the combined uncertainty. And this is one of the statistical uncertainty. Other uncertainties like non-statistical uncertainty. So let us say you have got the standard of lead or multi-element standards, and in which they have mentioned that this is like 1 ppm and 1000 ppm, and its uncertainty is 20 mg. So you have to calculate the relative uncertainty of that particular standard, which you have to calculate like this: error divided by its real concentration. So 20 is the error. So 20 divided by 1000. So this 2% is the relative uncertainty in that particular standard itself. So you have to gather all the statistical uncertainties and non-statistical uncertainties, and then you have to get the combined relative uncertainty. So this is the formula for calculating combined relative uncertainty. And then you have to get the expended uncertainty. So expended uncertainty is calculated like you have to multiply relative combined uncertainty with k. You know, k is the this factor. If it is k2, then you have to you are around 95% confident, and if you are having K3, then you are around 99.7% confidence. So this is the level of confidence. So expended uncertainty is accepted at K2 level. So all the results which you are measuring, which you are reporting, it should be below the expended uncertainty. So this is very important in the measurement of trace metal and in the isotope measurement. Until and unless you are not sure about your data, you cannot talk about all those steps which I have discussed in the earlier slides, like climate thermal analogs and all those things. So error will definitely incorporate in the data. So according to the International Organization of Standardization, laboratories must estimate uncertainty where relevant. So uncertainty must be consistent with method validation data. So all those method validation which we did before, like measuring ruggedness, repeatability, and repeatability, they should be less than the expended uncertainty. The standard deviation should be less than the expended uncertainty. We also need to do the inter-laboratory calibration. So how much of we are very confident that our labs do regular inter-laboratory calibration? Same standard we have to send to, let us say, ICER Pune or ICER Trivandrum, IIT Kanpur, or some other institutes where the same instrument is there, like IRMS or ICP-OES. And we have to check whether all the laboratories are giving the same data. So ILC is one of the important aspect of measurement of the uncertainties, and ISO 17025 laboratories. So there is no fixed limit to be imposed for measuring the uncertainties, but every lab have to make their own protocol and SOPs to report the uncertainty. And if the expended uncertainty is below 10%, it is excellent, and it is acceptable. It is if it is between 10 to 20%. So the reference for these two you can get from the Joint Committee for Guides in Metrology, guides to express the uncertainty measurement. Very detailed statistical and all those content you can get in these guidelines. So this is the most important part which I wanted to talk about in this talk. And with this slide, I would like to end my talk, and I welcome you all to ask anything which you want. Thank you.
Thank you so much, Dr. Tripathi. I think now, after having this wonderful, talk, and the focus was main, to know the how to calculate the uncertainties in the measurements. I think that is the most important aspect of our, chemistry. So now the floor is open, and I welcome all the participants. You can interact with Dr. Tripathi and put your suggestions and ask your questions. Please raise your hand, or you can directly write in the chat box.
Mhm. If you permit, I would like to show the instrumentation facility because right now I'm sharing the screen so I can show the instrumentation facility to everyone present here, what we have in SATHI BHU, if you permit.
Yeah, I think we can do. I think we can do.
Okay. So I think this is visible and these are the equipments which we have right now. So HR-ICP-MS, laser scanning super resolution microscope system, IRMS. This is NMR facility which we have here, and CD, circular dichromism spectrophotometer we have, photoacoustic imaging platform, and chromatography recently installed. HR-ICP-MS is also recently installed here. Then triple quadrupole LC-MS we have in SATHI BHU. So the purpose of showing this is if somebody is interested in these analytical instruments, so they can contact SATHI BHU and get their measurement done. And we are NABL certified, so there is no worry about the data.
So, like, how to approach if someone from some different institute have like basic idea and they want to measure the some kind of trace metal concentration or any kind of such analysis. So, how to approach and where to approach? Is there any particular online portal for that, or just like writing an email? If you can just uh, give a brief, like, one
Yeah,
minute discussion on
they
this.
can if they know what to measure and how to measure. If they know their if they know their protocol, they can directly contact SATHI and they can register here and they can send their sample directly. In which form they want to send, they have to mention everything. And if they want consultancy, they want to know that what to measure and what kind of interpretation they can do. We are open for that also. We can provide guidance that what kind of how to do the sampling and how to do the digestion, what acid has to they have to use, and all those things. It can be done very easily in SATHI BHU.
Thank you. So, I think if anyone have any doubt, any questions, please come front and uh, okay. So, Anand, you can just unmute yourself and you can ask the question if you are okay with that.
Size correction he is asking.
Uh, can you say something about size correction in case of
Size
nitrogen?
correction in the case of nitrogen isotopes. Size correction of what? You can unmute and we can discuss.
Hello.
Huh. Yeah, I'm listening.
Yes. So I'm from Birbal Sahni Institute, radiocarbon lab. I want to know if we are having a different helicot of a that standard of like sulfur or something, and we want to check the variation in the isotopic value with a change in the weight of a standard. So can you tell something about it?
No, you are saying you will take different weight
Helicot,
of
yeah.
this different helicot of the standard or sample?
No, different helicot of a sample simultaneously with a standard.
Okay, so ideally the isotopic value should not change with weight. So weight has
Like
nothing to do for the isotopic values. So if your samples, I'm talking about samples, if they are homogenized properly,
okay.
the value should not change. It will, let us say you are running three triplicates, you are running your sample in triplicates, they all should give you nearby same values. If it is not happening,
That's okay for carbon and sometimes sulfur, but in case of nitrogen, the size matters in case of standard as
yeah.
well.
So let us say you are preparing your tin foil capsules.
Okay.
It has some air remaining in it. So there is a we had in NCPOR when I was in the lab. We bought a particular specific instrument for making the tin capsules. So it presses it. There is a lever in that, and it presses your capsule and removes all the air from inside. So you will get a capsule like medicines, tablets you get now. So all the
Yeah.
air get removed. So you can try that, and you can run one sample triplicate and check whether the results are coming same or not. If you do it manually using your hands, or let us say you are wearing gloves and you are pressing it, sometimes some air remains in that, and nitrogen is present in the air, and you will not get the same result.
Okay.
If it is not clear, you can ask me more.
Okay. Thank
Yeah.
you.
I think Nishant has raised a hand. Nishant, you can unmute yourself and please ask the question.
Yeah, am I audible?
Yeah, I can listen you.
Yeah, thank you, Arvind, for starting this lecture series, and thank you, Subham, for presenting this nice talk. So I was interested on the thing that so you said that per degree Celsius there is incorporation of 10% of magnesium within the foraminiferal cells.
Yeah.
So whether this incorporation is similar for a particular species throughout different ocean basins, or does it have some regional influences in that incorporation?
Uh, just a minute. There's a paper. There's a recent paper. I'm just looking for that particular paper, because Leah has done this work long back in 1999, and she said that per degree Celsius 10% change. So she has talked more about Seglifer.
Okay.
But there's a recent paper by just wait. I can see that in my view. So your question is whether it is basin dependent, species dependent, or something like this.
Yes. Uh, so you said that per degree Celsius there is 10% of magnesium incorporation in foraminiferal cells. So whether it
Yeah,
varies for different species, or is there any regional influence as such?
it may vary, but not much. It may vary, but
Okay.
not much, because mg by ca is a we call it proxy because it is applied for can be applied for many different species. So let us say the recent paper which I was talking about right now, it has said that 8%
Okay.
change. So earlier papers they have talked about 10% change. So it varies like 2 to 3%. It can vary. Maybe some species is there in which if you do the culture study and if you vary 1 degree Celsius, maybe it will go up to 12% or maybe 7.5%.
Okay. Also, out of curiosity, I would like to know that what is the degree of variance or variability between this MGCA and TEX86, or even assemblies-based SST reconstruction? So, suppose for a particular reason we have all these three available, then which one we should be relying upon?
Yeah, so we have to check which particular empirical equation is giving you the less uncertainty, less error. And present day climatology, you have to check the present day climatology, what kind of temperature we are getting for that particular region. And let us say you are using three proxies and which two are agreeing. So uh, you have to kind of rely on the like t test or jd score you can take, and you can judge which particular proxy is better for which particular reason. But we know that all with the help of proxies we are trying to measure a particular parameter. So all proxies should give you the same parameter, same temperature or same particular parameter. If one proxy is giving you very absurd, then it should not be called as proxy. So nearby ही आएगा अगर आप लेटेस्ट से टेक्स 86 यूज कर रहे हैं और mg/ca यूज कर रहे हैं, क्योंकि टेक्स 86 ग्लोबली यूज कर सकते हैं हम. So from the same sample, from the same core, we can measure and then we can check whether how much variance is there. But I have not
Okay.
kept that for this particular talk. But that is one of my slides have talked about that. This one, that different paleoceanographic proxies capture different aspects of the thermal temperature variability. So we have to make sure that which proxy is reliable for a particular reason. It can be based upon your references, your literature survey, or it can be based upon your present day climatology.
Okay, thank you. And one more thing which I would like to ask is, so most of, as you said that your reconstruction was based on Saculifer. So did you
Yes.
find G. ruber in Arabian Sea? So why not G. ruber, or why you chose G. Saculifer for the reconstruction?
Yeah, I chose G. sacclifer because it was the only abundant species in IODP 355 core. G. ruber is
Okay.
very much, G. ruber is one of the most abundant in Arabian Sea, but in that particular core, only G. sacclifer was most abundant, and it is asked me many times that why I chose G. sacclifer and that
Yes,
particular
yes,
morphotype. That's
yes,
why I
yes.
have called it mixed layer temperature. I have not called it sea surface temperature because G. sacclifer will give you mixed layer temperature, MLT.
Yes, yes, yes. Okay. Thank you so much. Thank you for this nice presentation. So it
Yeah.
was quite easy to go through. And yes, thank you, Arvind, for starting this
Thank you.
lecture
Thanks a lot.
series. Okay.
Thanks.
Thank you.
I think we have one question in chat box from Geetika.
Yeah.
Geetika, you if you can, then I think you can unmute and discuss this. It will be better to communicate.
Thank you, Arvind Bhaiya, for this opportunity to um, attend talks online and to get to know people who are working in the same field. Dr. Shubham, it was a very good presentation. I worked on benthic
Mhm.
forams for my dissertation. I'm currently working in Indian Institute of Geomagnetism. But I would really like to know, how does this dissolution affect the Mg by Ca ratio in benthic foraminifera? How will we calculate it? Like, how can we be certain if
Okay, so
dissolution is?
I would like to know something from you. You have already worked on Mg by C of benthic forams.
No. I didn't work on isotope. I have worked on the abundance of the diversity of the
Abundance.
benthic foramin.
Okay,
Yeah,
okay,
yeah,
abundance. Okay.
yeah.
So benthic forams are very, rugged, you can say, compared to planktic forams. They are not as prone as to dissolution as planktic forams are. But
Mm-hmm.
if you see the variability for the bottom water, it is very less.
Okay.
So on the surface or on in the mixed layer, as I have shown in the graph, the temperature variability can be 2 to 3 degrees Celsius. And if you go from equator to pole, it is around 10 degrees Celsius. This variability on the surface, but in
Mm-hmm.
bottom water it is very less. So how dissolution affects Mg by Ca? Dissolution will affect your carbonates. You will not get the real carbonate which forams has precipitated in the real climate or real world. So if
Mm-hmm.
dissolution is taking place, it will remove the carbonate, and if carbonate gets removed, Mg and Ca and many other things will get removed. So we can say that if the forams are not in their real form or not in their intact form, it will not give you the Mg by C of the real forams. You know what I am
Okay.
telling?
Mm. Yeah, I am getting it. Okay.
So
Okay.
for benthic forams, I personally don't consider Mg by Ca as a very reliable proxy because we have to have a range of temperature to get the you know variability in the
Mm.
temperature. If the range
So
is
on
very less,
benthic
how?
forams, so on
Uh-huh.
benthic forams, what can we actually work on? Like, what if not Mg by Ca, then what?
Benthic forams, there are other specialized people for that, but as far as I know, you can do something which has glacial and interglacial variability. You can reconstruct circulation, bottom water circulation, and you can go for research in water mass variability and all those things. But there are people
Mm-hmm.
who had worked extensively on benthic forams, so you can share here, or we can have a talk on those things in future. So
Mm.
glacial and interglacial variability is very reliable by using benthic foraminifera proxy.
Okay. Okay, got it. Okay.
Thanks, Geetika. Anyone else who wants to have an interaction?
So one thing I want to discuss here, because around 19 people are here. So after seeing the slides on uncertainty measurement and error measurement and validation method, how many of you are very confident that your instrument regularly goes through that process? When was the last time your instrument was calibrated? Let us say IRMS or ICP-OES for all these particular things which I have shown in my slides. what is the expended uncertainty for your instrument. This is just for discussion.
I had a similar question actually when I was seeing that slide. Uh, what happens if we use different standards, let's say VBDB? If we're using a different standard for calibrating every other time, will the instrument, what will have effect? What will be the effect with the values if we use
No,
different standards in
as I
different
said,
states?
when I was discussing with Nishant,
Mm.
you are not measuring standard, or you are not measuring a particular proxy as a chemical or something. You want to measure the climate change, you want to measure some particular parameter. So all those standards, they should ultimately lead you to the realistic climatology or any parameter. Let us say if I have two different standards for measuring mg by ca, so all those two different standards should give me the same temperature for my samples. If they are not, doing that, they are not called as standards. Got it?
Okay. 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 Mm.
Yeah, so in this regard, I think I want to add few things that many of here, like sometimes they don't get opportunity to work directly on the instrument, as there is a limitations of using the instrument. Some like we mostly depend on sending the samples, and then the technical person is measuring the trace metal or anything for us. So just keep in mind that before taking your data, just do all kind of corrections, check the blanks, check the uncertainties, repetition, precision, accuracy, everything, and then do some manual corrections. There are a proper methodology for that. And once you get sure, then check your data with the published data from the regions also, so that something abrupt is coming or not coming before interpreting any abrupt data. Do all this exercise, and once you get sure that okay, now my data is fine, my blanks are okay, my standards are fine, my corrections are done, I think then you uh, start interpretation, and if something
Yeah.
is not coming in between, then you should discuss with the technical staff
Yeah,
who's
and
handling that.
one more thing, Arvind, we should also not get biased from the previous data, previous published data also. If you are very much sure about that our instrument is very reliable and all the uncertainties you have calculated, then whatever data you are getting, you must stand by that data. Whether a lot of big
Yeah.
peoples or big scientists have already generated and your data is getting means diverging from that already published data. But if you are very sure about your instrument, if you are very sure about your process and all those things performed in the lab, you must stand by your data, you must defend it.
We have one more question by Dr. Ped, I think Dr. Peduma, and he had mentioned if you can read the chat box, please.
I think it is a kind of comment
It was
from Pradyumn. It is a comment.
thank you.
But the purpose of this talk was only to show the method validation and how to calculate the uncertainty.
Yeah. So I think the purpose of the talk is, I think now like most of the people, they got to know how to work on this. And the presentation, this talk will, I think I've recorded the talk and it will be uploaded on YouTube. So the later on you can go and watch this talk again and again if you want to discuss something. I will also share the slide with the permission of Dr. Tripathi to all the participants and all our Paleo ECR India community members who have registered themselves on our platform. And then you can directly reach out to him. And he's currently, I told that he's working in a SATHI BHU, a very in one of the finest, instrumental facility in India right now. So I think one of the you can utilize this facility for a future, measurements and other things. And you can also discuss any kind of technical things to him. And with this, I think I should thank Dr. Tripathi for his valuable time and for discussing all this, technical and very fine things with all the earlier researchers working in the field of paleoclimatology, especially in the temperature measurements. And thanks everyone for joining. Thank you so much.
Thank you, Arvind, for this platform. Bye bye.
Bye bye.